Well, we might have saved the best for last here. Our next presenter is Karen Mock. She is a molecular ecologist in the USU Wildland Resources Department. Her research questions are about gene flow patterns, inbreeding, and genetic divergence among populations in natural landscapes. She works on a wide variety of taxa, including aspen, mountain pine beetle, freshwater mussels, leopard frogs, bears, grouse, suckers, trout, and turkey. So, welcome.
Can you hear me out there? Okay, good. Thank goodness for microphones. So I'd like to start by acknowledging a couple of co-authors here. Paul Wolf, who is with the USU Ecology Center in the Biology Department, and also John Shaw. And there are actually many, many people who have pitched into this research. I'm summarizing lots of things that have been done over time here. Okay, oh, pointer, okay, good. Okay, so I'll start with some slides that you've seen before, and before. But maybe I can add a little bit to this. As Jim really said this, Jim Long said this very nicely, aspen has this enormous spatial and ecological amplitude. It goes all the way from essentially sea level to tree line. And it does that over this gigantic range from Alaska to Newfoundland and down. And people kind of say, yeah, down south. But look, I mean, we're looking south. This is way south. You can find aspen in central Mexico. It's a riparian species there, which is really interesting. So it's a huge, huge amplitude. It's a good focal species for a topic on resilience. So aspen regenerates, as you well know by now, in a couple of different ways, as it turns out. It can regenerate by suckering or asexually, and it can also reproduce by seeding, and surprisingly often, apparently, in the western part of its range by seeding. There's another familiar slide. This is such a pretty slide, and it's the kind of thing you think about, at least I do when I think about clones. So if you have aspen reproducing by suckering, but it originally started with a seed. It started with this little tiny thing that's the size of a grain of pepper, a little grain of pepper. And it grew by suckering to something that may be as large as that. Now, I think Jim used this slide to illustrate that we might have an oversimple view of clonality and maybe the size of clones in aspen. We haven't tested this. I would look at that and say, yeah, that probably is one, maybe there's probably another. But I think the mistake we've made through time is that it's that third one. It's easy to pick out the first one. Yeah, there it is. Oh, yeah, there's another. And then we've done something. And so the surprise has really been that this gushy stuff over here is probably many, 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 many clones, and that's been surprising. But our eyes are always attracted to these boundaries, aren't they? So a clone here, I'm going to use that term instead of the word genet. I'm going to try to use that word instead of the word genet. But by that, I mean multiple trees of the same genotype. And they originated from the same seed, all those trees that are genetically essentially identical, and they expanded through suckering. Now, don't run out the door. My lab has been doing a lot of work on aspen over the past 6 or 7 years using genetic markers. And I am going to take a little bit of time to tell you a tiny amount about these markers. The markers that we use most commonly for these kinds of questions are microsatellites. They're called microsatellites. And you can use, this is just a type of marker system, you can use those to distinguish individual clones. You can use them to distinguish half-sibling groups or identify parents. You can also use those to look at genetic similarity over landscapes. And so they're a really, really handy and extremely powerful tool. But I do want to tell you just a little bit about how they work, because it was the use of microsatellites that led us to our discoveries about triploidy. So microsatellites are these little locations on the genome. So think about the genome, all those chromosomes hanging around. You are a diploid, which means you have one copy of your mother's chromosomes and one copy of your father's chromosomes. Aspen, mostly, are also diploids. They have a maternal and a paternal set, and so they've got two of each copy of their chromosome, so two of each chromosome. Microsatellites are these markers that are scattered all over the genome, and if you look at one particular microsatellite, you're looking at that location on a chromosome. But it's a diploid, so you're going to find, you could potentially find two different versions of that marker. Microsatellites are just a little piece of DNA. They vary not so much by sequence as by length. So these are length variants. Now, so this is what microsatellite data looks like. There's a pair of peaks right there. This is one locus. This is just one little location in the genome. And each of those peaks is a different size. So we know that one parent of this organism, I put up here that it was a tree. I just made that up. I don't really know what that is. But these two versions, you have one peak here, and it is at 249 base pairs. That's how you measure the length. You have another peak here that's pretty close, 251 base pairs. So you know that this tree got one version of that marker from its mother, and the other version it got from its father. Okay? Now let's look at tree number 2. Tree number 2 also has 251. It has the same size, right? But it also has 258, and it lacks 249. So again, tree number 2, it's got nice, well-behaved diploid. So it's got two versions here, 251 base pairs and 258 base pairs, and that's what the data looks like. That's how I would score that data. Okay, tree number 3. Now, what's going on here? Tree number 3 is still a diploid, but both versions of the mother's versions and the father's versions of the chromosome have the same size. They have the same allele. We use the term allele to mean a variant at that particular locus. So this guy has 2 alleles. Tree number 2 has 2 alleles. Tree number 3 has 1 allele, but we know it's got 2 chromosomes, so it's the same size on both chromosomes. Does that make sense? Is everybody with me on that? Okay. Tree number 4 here is another one. It's got 2 different alleles, one from the mother, one from the father. If you look at the sizes here, depending on where you found tree number 4, it has 249 and 258. Well, this could be the progeny, right? It could be the offspring of tree number 1, which could have donated 249, and tree number 2, which could have given it 258. Everybody with me on that? Okay. I know I'm not talking to geneticists here, so give me the look of terror if I need to slow down. Okay, so those are the kind of markers we use. They're extremely variable. They're extremely variable. We use not just one of these per tree. We might use a panel of 7 to 10 to 12 to 20 of these, depending on our question. And so you can imagine the statistical power that we've got. It's huge. We can really tell individuals apart. So that's what we do. So I'll jump right from that, and I'm going to come back to that slide, sorry to say. I'm going to jump right from that to this picture. So we used microsatellite markers to survey aspen across its range. And we found, using those markers, that there is a big and obvious genetic subdivision that occurs right here in its range. So we call this the southwestern group, and we call this the northern group. And that blue line, you may recognize it, is the last glacial maximum. Okay, that's interesting because it means that it's very unlikely that the trees that are now in the southwest actually were the ones to recolonize after the glaciation. Those guys must have come from somewhere else, maybe various glacial refugium. Okay, so that's kind of an interesting finding, and it raises this question, what do we call these? I never know what to call these. I call them clusters. But some people might call them subspecies, they might be called races, they might be called ecotypes. We're not really sure what to call them right now, but they're definitely two different groups. Okay, but that's not why I'm giving this talk. It's kind of a nice piece of background to the rest of what I have to say. While we were doing this work, oh, and I should point out here too, the continental divide comes right through here. I should have put that on this map. And you'll see that in a second again. So back to this slide. While we were gathering the data for this big, big study, we ran across something that just drove us crazy for a while. And it was this, where's my pointer? Oh, there it is. This pattern. What? So I just explained about diploids, and you get one kind of allele or two, but we were seeing three. And we were seeing three repeatedly across lots of different loci. And of course, our first thought was, oh, we've got contamination. So, you know, we re-ran the sample, re-extracted the sample, do it again, clean the lab bench better, do it again. And we kept finding it, three. That led us to believe that we might have an awful lot of triploids. And so we began to ask questions about triploidy. So first I have to define this. You know what a diploid is, hopefully. two copies of each of the chromosomes. With triploidy, you've got 3 of each chromosome. 3 different copies of each chromosome. 2 come from one parent, and 1 comes from the other. And I'll give you a mechanism here in a minute. But that's what I mean by triploidy. Now, before I go into how that might happen on a cellular level, I want to go to this. Why is it? Who cares? So what if there's 3? Isn't 3 better than 2? There are a few reasons why we might care about this. First off, in aspen that are triploid could be more vegetatively vigorous in various ways, and I mean to be vague about that term, vegetatively vigorous. But in other plant species that demonstrate polyploidy, they are often more vigorous in one way or another than the diploid counterparts. We see it in lots and lots of different plants. So there's that. It would be nice to know if there was a certain kind of aspen out there on the landscape that was more vigorous in one way or another. Nice to know that. Another interesting aspect about triploids, because they have 3 copies of their chromosomes, they can do just fine vegetatively, and they may be doing especially well vegetatively, but when it comes time for them to make gametes, and they try going through meiosis to make gametes, and they've got 3, it doesn't work. And so when they try to go through meiosis, they don't make gametes very well. So they're either going to be completely sterile or have very, very low fertility. That might be something that you might want to know about. So that's another reason. In addition, triploid plants and all polyploid plants are expected to have bigger cell sizes. And if you have bigger cells, that might influence your physiology in a variety of ways. And one of the most immediate ways could be xylem elements, for example. So, I mean, this is just a kind of a thought exercise right now. We're doing this in the lab eventually. But if you have larger xylem elements, if you're a triploid and your xylem elements are larger, you might be able to move water really well, right? That might be good in certain circumstances. On the other hand, you're going to be susceptible to what? Cavitation. You're going to get air bubbles in those tubes under drought conditions or under freezing conditions potentially. And you might undergo cavitation more easily than if you had smaller xylem elements. That's a really interesting thing. So they could do very well in certain circumstances, but then when they die, if they undergo drought or freezing, they could cavitate more rapidly than the other kind. So we're checking that out to see if that's true. Okay, but this is a bunch of reasons why triploids might be interesting. There we go. Okay, so I do want to This is another one of those Bio 101 from your nightmares kind of slides. But I wanted to mention how triploids arise because there are a lot of misconceptions out there. In the plant world, most of the time, triploids arise from something called unreduced gametes. And it happens, so let's say you have a normal, well-behaved diploid tree, and it undergoes meiosis to make gametes, to make sperm or egg, right? And so it undergoes meiosis, and normally you undergo meiosis and you produce haploid gametes. They just have one copy. You get two haploid gametes together, put them together, and you have a zygote and a new individual. So that's how it's supposed to work. But meiosis can make mistakes sometimes, and when it does, you can end up not with haploid gametes, but with diploid gametes. And if you do, then you could end up with a diploid sperm or egg, which then unites with a haploid sperm or egg, and now you've got a triploid. And that's how they're supposed to be. That's how it happens most of the time in plants. Now, an interesting side note here, is that, keep covering that up. You expect, this kind of accident in meiosis is pretty rare. So you expect the formation of unreduced gametes to be pretty rare. You expect that to be low frequency. You also expect the proportion of triploid progeny in a batch of seeds to be pretty low. You don't expect to see very many of those. Okay. I think I'll not talk too much about this. This was just a slide on a couple of different ways to detect triploids. We can detect them with microsatellites, like I said, and we can also use nuclear density and something called flow cytometry to look at differences, diploids versus triploids. You can also, if you have the, unlimited time and money and patience and don't mind migraines, you can actually do cytology. You can do chromosome squashes and actually count the things. Okay, so there are different ways to do it. Okay, so here's the interesting part then, I think. How is triploidy distributed at local spatial scales? This is a slide that you saw in Jim Long's talk. He was using it to illustrate clonal diversity. But what he didn't mention here is that the black points here, each symbol is a different clone. The black points here are triploids. Whoa. Now, this is a way higher frequency than you would expect to see in a seed crop. Way higher. It's really weird, and we had checked this every way back and forth you can imagine, because we were sure we were wrong. So the black symbols in here are triploids. So, first off, some observations. There are many triploid clones present, way more than you would expect in a seed crop, which suggests that they're being selected, or have been selected somehow in the past. Another observation here that you can kind of see in these patterns is that the bigger clones are triploid. This is actually a very strong pattern. When you have big clones, they're almost invariably triploid. Some of the triploids are little clones, but those big ones are almost always triploid. Also, and this is a funny one, a bizarre one, triploid clones cluster spatially. We've seen that in not just this study site, but a couple of other ones too. So they cluster spatially. Why might that be? Oh. So this has led us to a whole bunch of other questions, as you might imagine. This is a little bit of preliminary, it's data from a student of mine, Richie Gardner, who will publish this very soon. And so this is, he compared diploids and triploids in that same study area and found that the triploids had significantly higher chemical defenses. These are phenolic glycosides, which are famous for deterring herbivory. Okay? So that's something to put in your hat about triploids. We don't know if that's true of all age classes, or whether it's true in other landscapes, or whether it's really correlated with a less ungulate browsing pressure. But those are a whole bunch of very interesting things to ask. Okay, so we couldn't resist. We had just done this big range-wide study using microsatellites. So we asked, and we published this in a separate paper, but we asked what triploidy looks like on a big landscape. We knew about the little landscape. Now, what about the big landscape? Well, this, I don't know if you can see this very well. These pies here represent the proportion of triploidy, the red being triploids. The take-home message here is that this this southwestern cluster, which is different genetically, is also tends to be much higher across the board with respect to Triploidy. We have a couple of exceptions right here. One of the exceptions is in Yellowstone, POTR Yellowstone. Another one's down here in Arizona. These are both batches of seedlings that we tested. They have weirdly, for the West, weirdly low Triploidy, totally what you would expect from a seed crop. So that was kind of cool to find. So these, we have really high Triploidy in the West, very low Triploidy here. Again, glacial maximum. This time I remembered to put the continental divide in there. And so that's kind of an interesting pattern. Okay, this roughly corresponds with climate. And you probably already knew that because you know the climate of that area. I'm not going to spend too much time on this one. But here are a bunch of questions about Triploidy. And I wish I had answers to these, maybe in a couple of years. Do they have faster shoot growth? Do they spread faster by roots? Those might be totally different answers. Is there less herbivory? Do they use water more efficiently? Are they more drought tolerant or are they less drought tolerant? And again, Triploids might not only have specific strengths, but they might also have particular susceptibilities to drought. So those are all questions that we need to answer. Now, a little bit about management implications of this. So in terms of management, there are two kinds of regeneration that you think about managing through, I guess. One is by coppicing, and the other is by, one way or another, encouraging seedling establishment. These might lead to very different kinds of forests in terms of Triploidy. If you're doing coppicing, then you're going to be favoring asexual reproduction because you're causing the clones to just persist and expand. That's what you do. That's going to favor Triploids if the big ones are Triploids. Okay. If you regenerate by coppicing, it's not going to allow you to establish aspen in new areas. In addition, it doesn't allow you to do sexual recombination, which leads to genetic variation and adaptability. And it leads to the loss of genetic diversity over long periods of time. Okay? By contrast, if you regenerate by trying to encourage seedlings, you're going to favor diploids because the seed crops are going to be mostly diploids. You're going to favor sexual recombination, favor genetic diversity, increase the evolutionary potential, and potentially you could, let's see, well, I won't go there too much, but the seedlings, if you have a lot of seedlings, you're going to have a dependency on the window of opportunity for the right soil conditions to germinate the seedlings and to have them persist. In addition, you might have what you could call a window of susceptibility for seedlings when root systems are getting established and the stems are small and grazable. Okay, so that was a bunch of questions. Hopefully that's some future research questions. We are looking actively at a few of these questions right now. I have two undergrad students. This is Brianna. She's so proud of her leaves. And this is Abby. These are two undergrad students who were brave enough to decide to work with me on some of these. They're looking at, first off, stomatal sizes. So you can paint fingernail polish on the back of an aspen leaf and make an imprint, look at it in the microscope and measure stomate sizes. How cool is that? So that's what she's doing to see if stomatal sizes are different between diploids and triploids. If they are, we've got a really nice field method for distinguishing diploids and triploids without all the genetic trouble. And we're also looking at diploids and triploids and drought cavitation susceptibility and also growth and physiological differences both in the field and in common gardens. Okay, so hopefully I'm not too much over. I've got lots and lots of people to acknowledge. I'm not going to go through all that. And we have kind of scrapped together lots of little bits of funding here and there to make this work happen over the years. So I hope you have some questions for me.
That we might be carrying nail polish in our cruising vests?
It's not, you have a special container. Yes.
Did you notice that certain loci were more common in the triploid populations?
All all of the loci are there regardless, the loci are. What we did notice that there was a very strong tendency if you were triploid, if you had 3 alleles at one locus, that you were very likely to have 3 alleles at many other loci, which was more evidence that this is a genome-wide thing. Yeah, good question. Oh, not directly. It's a little hard with triploidy for technical reasons to really count the alleles. Well, I'm not going to go into that, but it's a harder question to ask than you might guess, and I'd be happy to email with you about that. Yeah. Yes, Chris.
Do you think that the glacial retreat led to more seeding in those populations, and that led to the diploid pattern, or do you think the environmental differences led to their reproductive style and that led to?
That's a really good question, and I don't know. My sense is that, that you have a lot more seeding going on in that northern range, and that maybe that's, the hallmark really of the glaciation. On the other hand, there could be something about that southwestern group genetically. They might produce unreduced gametes a little more often. Yeah, so it's hard to say. There have been papers out there that suggest that there are environmental conditions that trigger the production of unreduced gametes. It'd be a cool thing to look at. Yeah.
Yeah. So do you think that under future climatic scenarios that diploids will become more common in the West, and therefore successful regeneration from seed will become more common?
I guess it depends on which climate model which climate projection you're looking at. And I really can't answer that unless I know what these things are adapted to, and we just don't know that yet, to be honest. All we know is that the triploids are big, or the big ones are triploids, I should say. And that suggests that they have some advantage. Maybe they spread faster, grow faster, maybe they're just older. We don't know if that suggests that they're going to be more successful in the future or that they have been more successful in the past. I mean, it's entirely possible that some of the large-scale die-offs we're seeing are big triploid clones going down. I mean, that would be a really interesting thing. So I don't know that yet. I wish I did. Yeah.
You mentioned that there are some advantages physically to having a triploid pattern. Did you see any resistance? I mean, do they seem to be generally healthier to fungi?
Every time I think I see a pattern in the field based on the few that I see, it's almost like a joke. You go to 3 or 4 and it's like, yeah, triploids have the big waxy leaves and they're resistant to this kind of fungus, and the next one you see has little wimpy leaves and has that fungus all over it. I haven't seen enough of it. I haven't done a study that would allow me to ask that question. Is really the answer. I wish I knew, but at least now we know the question to ask, which is a really cool and different thing. We never knew to look for triploids before.
Farthest extent on your map, the farthest south?
Mexico. Yeah, we got, yeah. Yeah, they're mostly triploid there, those little riparian things. I should have brought a slide of this. But, yeah. Yes, Jim?
Just a comment. If as a silviculturist, if I'm convinced that I want to manage with triploids in terms of establishing them in areas where they don't exist, I mean, we can do that with rooted cuttings. So I mean, that kind of assisted migration.
Yeah.
It's not a barrier.
Yeah, you could do that with cuttings. It would be a long haul, but I think you could do that with cuttings. You might also be able to accomplish it with seeds if you have a low proportion that are triploid and selection is strong and directional. You might actually be able to pull it off even with seeds, but it wouldn't be as efficient. Somebody back there was very patient. Yeah.
I'm curious if you think that the apparent limitation of seedling establishment in the western aspen, do you think that has more to do with the lack of seed?
The apparent what? I'm sorry.
The lack of seed.
Oh, the lack
or the abiotic factors.
Oh. So are you asking whether there might be a lot of triploids out there with bad seed?
Right. Well, I think there's a lot more seed in the northern population.
Yeah, there's a lot, but the reproduction by seeds is much more common. And the clones are much smaller, and so it's not nearly as unusual to see batches of seedlings in the northern populations. Yeah.
It's a function of the triploidy versus diploidy.
Well, I've actually wondered that, because if you have triploid, if your landscape is dominated by triploid clones, let me go back to this map really quick. If you look at this map, this is the proportion of clones, sorry. This is the proportion of clones that are triploid versus diploid. If you add to that the fact that most of the big clones are triploid, the proportion of trees that are triploid is huge. It's just huge. And if they're putting out seeds that don't work or gametes that don't work, you know, you could have some problems there. I think it would be a very interesting thing to look at. Yeah.
I have two questions. In looking at this pattern from a biogeographical standpoint, do you have any genetic evidence that can suggest place of origin for each of these, the diploid or the triploid individuals?
We don't. We thought the classic way to look at that genetically would be to look at areas where you have unusually high diversity with this kind of data. And it was kind of the diversity across that northern range is smeared out. There's not, we weren't able to, in our data at this scale, to find pockets of diversity. We may have missed them, but we tried to sample where known glacial refugia are. We just never saw the pattern of increased or decreased genetic diversity across there. They're all kind of the same.
Then in either of these.
I think some of the upcoming genetic tools, some of the next generation sequencing kind of stuff, if you're familiar with that, might help us with that. But we sequenced some of the fastest mutating chloroplast genes we could find, and we couldn't find any differences. It was really surprising. This is really recent.